Good afternoon, and thank you very much once again for the kind invitation and for having me here today. So my name is Konstantinos Gerasimidis actually, and I work and live in Glasgow for the last 20 years. But as you can see from the looks and from the accent, I don't come from Glasgow. I come from Greece. And as you probably know, Greece and Saudi Arabia share many things in culture and in food. So my presentation today is to talk about diet and how diet interacts with the gut microbiome. And we'll focus mostly on pediatric gastrointestinal diseases. And we do this because they asked me to do that. But more important, because this is an area of personal research. I work in Glasgow, which is the fourth oldest university of the English-speaking world, and I run a very large research team which lies between basic science and clinical medicine. So I hope you all agree with me that pretty much everyone loves the microbiome, and the previous talk was probably the best introduction I could have. There's a lot of interest. When I moved in Glasgow in the beginning of the millennium, no one really cared about the microbiome. I was the only one around the hospital who was going to collect stool samples from children with IBD. But as you can see, our interest has evolved exponentially over the last decade, and I think this trend will carry on and will not plateau, at least for the next decade. And you may wonder why this has happened, why have suddenly everyone is so much interested about the gut microbiome. And I think the main reason behind that is that the way we understand the etiology of non-communicable diseases has evolved a lot over this period. So back in 2000, we were very much interested about the genes, and now we're talking about monogenic and polygenic disease. But even if you try to put all the genes which have been associated with risk of Crohn's disease in the same pooled analysis, you only explain pretty much 20% of the risk of people getting IBD. Okay, so IBD, for example, is not a genetic condition. Of course, the paradigm shifted. We focused in the gut microbiome, but again, in this area, we haven't managed to identify certain pathogenic or pathobiont organisms which explain, for example, why some people get IBD, why some people take type 1 diabetes, and so forth. And we are now in 2023, and we believe that all these three factors are really important, but all of these factors are important under the influence of environmental factors. This is something that we often call exposome, and among the most important environmental factors is the diet. And one of the things we tend to forget is that when we actually have food, when we feed our body, when we give our body nutrients, carbohydrates, proteins, fat, fiber, what we actually do at the same time is to feed our gut microbiome. And I would like you to remember this, because this is very, very important in understanding the biology underlying various of these non-communicable diseases. And this is a very intimate relationship, and essentially what you eat pretty much defines what your microbiome will look like, and also defines the composition, but also the functional traits, the functional ability, capacity of this microbiome. So this is one of the seminal papers published by the Italian group, the De Filippo. And what you can see here, without going too much in detail, is that if you have a diet which is rich in vegetables, in crude grains, this will create a very different microbiome compared to the microbiome that Italian children have in the modern Italian kind of like region. So these children tend to eat more pasta, more pizza, and as you can see again from this slide, there are different compositional shifts. So what you eat pretty much determines your gut microbiome, but not just the composition, but also its function. And on the right-hand side of this slide, you can see some of the most important metabolic products that the bacteria produce following fermentation of indigestible carbohydrates, in other words, fermentable carbohydrates, fermentable fibers, and these are the short-chain fatty acids. So acetate, propionate, butyrate are important. We want to believe that butyrate is the Justin Bieber, is the most important of all these short-chain fatty acids because it has been associated with protection of cancer and other inflammatory conditions. But of course, in our diet, we don't only consume nutrients. We don't just consume carbohydrates, proteins, and fat. Our diet is very much industrialized, so we start introducing in our food chain food components which do not have any nutritional value, but are very, very important in order to make our food look better and to make our food to last longer in the host, in our cells and so forth. And there's very little research on how these food additives can actually influence the host, our human body, not so much in terms of the acute effects, because we do that, but with regards to the long-term effects. So this is one of the papers which was published by Benoit Sasseng in France, and essentially the authors claim here, using an animal model of colitis, that certain food additives, certain food emulsifiers can cause dysbiosis, can increase the proximity, the contact essentially of the gut microbiome to epithelium, initiated inflammatory response, and of course causing colitis. Now, this talk is about pediatric gastrointestinal diseases, and last year we were commissioned by the Royal College of Pediatrics in the UK to write a very simple review article which is aimed to pediatricians. So if you want to know a little bit about the role of the gut microbiome in various GI conditions, I think this is a good read for you. But as you can see on this picture here, we know that in many of these non-communicable diseases, like, for example, inflammatory bowel disease, like irritable bowel disease, intestinal failure, and necrotizing colitis, there's a lot of dysbiosis. And what does dysbiosis mean? It means essentially that you've got different bacteria to the healthy people, that your gut microbiome has a lower diversity, and you've got some pathobionts, some bacteria which can be nasty under the right conditions, which are high predominance and high prevalences, and on the other hand, you've got some other beneficial organisms, like the bifidobacteria, which are at much lower concentrations. Now, when you see slides like the previous one, you think, okay, we have done a case control, a cross-sectional study. We found differences in the gut bacteria in a disease compared to the healthy status. And the obvious hypothesis to make is that, this is the reason why these people get IBD, why they get IBS, why they get necrotizing colitis. But I don't think it's always important and it's always right to blame the gut bacteria in the onset of pediatric gastroenterology. Gastrointestinal diseases, apologies. So when you come across this data, I would like you always to take this data with a pinch of salt, and you have always to consider the various caveats which associate with data like that. As I told you before, there are quite a few in vitro and animal-based studies which say that food additives are bad for you, but at the moment we hardly have any dietary interventions to show that this is the case. And actually the latest 2 dietary interventions using diets which excluded food additives in Crohn's disease have shown no benefit. Okay, so it's very, very important before we start making recommendations for patients of how diet can affect your microbiome and disease progression, it's very important to translate this animal data to human trials. Of course, quite a lot of the claims, the hypotheses originating about the role of a gut microbiome causing disease come from cross-sectional studies. And the most important drawback with these studies is that you cannot address reverse causation. So in other words, it's the chicken and the egg story. So is it the microbiome which came first and it was disturbed and because of that you develop inflammatory bowel disease? Or is the fact that you actually have inflammatory bowel disease and because of all the consequences that this disease has on your body system, this can actually change your microbiome. And if you think in the context of gastrointestinal diseases, as you can see from this slide, bottom right corner, when you have GI diseases, a lot of things are happening in your body. Your gastric time is changing, the pH is changing, your immune system is changing, you start giving patients different types of antibiotics, drugs, you give them x-rays, the patients change their diets, okay? And all these things actually can change the microbiome. So it's very important to think about that. And the last caveat here is what we call residual confounding factors. So basically, we explore the association between exposure, in our case, microbiome, and an outcome, in our case, pediatric gastrointestinal diseases, how much of these associations can be explained by other factors. Now, thinking about the role of the gut microbiome and how it interacts with diet in the development, treatment, and progression and outcomes of patients with pediatric GI disease, I would like you to focus on this slide. So this is what we call the causal pathway. So we want to believe that microbiota causes gastrointestinal disease, but in this kind of like pathway, diet can be a confounding factor, as I told you before. And there's a very nice study which was published last year in Cell saying that all the changes in the microbiome of children who are autistic are explained by the dietary patterns of autistic children. Okay, so really good data to suggest that microbiome doesn't cause autism. It's the fact that when you have autism, this is associated with changes, with radical changes actually in your dietary habit, and these changes change your microbiome. Now, another important role of the gut microbiome and how it interacts with diet is that we need our microbes to get the benefits from some of the food. For example, fiber is really important, okay? And the fiber is really important for two reasons. If it's not fermented, because it gives stool bulk and helps with defecation, but if it is fermented, then it's broken down by the gut bacteria producing short-chain fatty acids, okay? So you can see here very nicely one of these examples. And of course, the most exciting area for me is whether we can actually use diet in order to improve the clinical outcomes of patients. Can we actually use diet to modify the microbiome in order to make our patients feel better and so forth. So I will very, very briefly go through some of these studies, and this is all studies which have been carried out in my department in the University of Glasgow. So there is a lot of interest about celiac disease. There's a lot of interest about gut microbiome causing celiac disease, because we know that the first exposure to your body to gluten does not really tell you if you're going to develop celiac disease. So there must be an environmental factor. And this is a paper. But when you think about this association, you have also to think about reverse causation. Okay, so when you have celiac disease, you have malabsorption, you have changes in GI triazine type, and all these things change your microbiome. And then when you treat celiac disease, as you can see, you introduce in your diet a very different diet. You exclude cereals, and of course you exclude fiber, and of course this can improve your symptoms, you can make you feel better, but at the same time can also affect your microbiome. So this is a study that we published a couple of years in gastroenterology, where we try actually to address these causal effects, these confounding effects. And without kind of like going in much in detail, we have identified the majority of the species which differ in patients with celiac disease, differ because these patients follow a gluten-free diet, which is a diet which has a very different composition to the normal diet. But we also identified certain species which were always a distinct microbial signature of patients with celiac disease, and we think that these are very important species to use as disease biomarker. We also found that butyrate is very low in this population, and it does make sense because these children avoid cereals. And for us, this has clinical implications, which mean that actually you may want to encourage our children with celiac disease to consume non-gluten containing alternatives of cereals. The second study I will try to discuss with you is for patients with salt bowel syndrome, with intestinal failure. This is work I have carried out in collaboration with the Erasmus Children's Hospital. And you can see from this slide, well, when you have intestinal failure, you have probably the most extreme example of microbial dysbiosis. You can see your diversity is changing, you can see the composition is changing. And when we're looking at beneficial species and beneficial microbial metabolites, as you can see, they're totally different, okay, to the healthy status. On the right-hand side of this slide, you can see that the most dominant bacteria in children with intestinal failure receiving parenteral nutrition are Chronobacter, are E. coli, are Staphylococcus, okay? These are very subdominant species of the healthy human gut microbiome, but in these children, these species are really in high abundance. And the question is, what does it mean? And in our eyes, these are very important signatures. We know that they may be caused because of how these children are treated, but there is now evidence to suggest that some of these actually microbial disturbances can be important in the onset of comorbidities that these children have. They may be, for example, important in development of parenteral nutrition, fatty liver disease, in D-lactic acidosis, in small bowel overgrowth, and this is something that we are currently studying in my group in Glasgow. Now, the last example I would like to talk to you about is IBD, and this is our bread and butter in Glasgow. This is where I do most of our research, and there are a lot of papers like this telling us that the microbiome of children and adults with inflammatory bowel disease is very dysbiotic. But of course, the question is, which came first? Is it the dysbiosis which caused IBD or vice versa? And the best way actually to address that is to think about microbial, pharmacological, or other ways by which you can manipulate the microbiome of these children and try to see what is happening if you do that. So in other words, do patients get better if they change the microbiome? And this is a figure that we have included in these review articles, and these are the various ways you can do that. So if you want, for example, in your microbiome to suppress it, you can use antibiotics, you can use exclusive enteral nutrition, you can use certain type of dietary therapies. And on the other hand, if you want to cause eubiosis, if you want to have a healthier microbiome, you can use prebiotics, symbiotics, probiotics, and so forth. But the question, how do this perform in patients with inflammatory bowel disease? And this is really fascinating because we now have data to suggest that in patients with Crohn's disease, when we try to normalize their microbiome, all of these interventions have failed. In other words, prebiotics do not work, probiotics do not work, okay? But what works is the opposite. What works are interventions which aim to suppress the microbiome in these children, like antibiotics, like exclusive enteral nutrition, and so forth. In ulcerative colitis, we have actually the opposite extreme. Here, all these factors, all these therapies which aim to recolonize, to improve, to restore our dysbiosis work, whereas all these other interventions which aim to suppress the microbiome differ. So this is very important data which obviously has very important implications. Now the last slides will be about options to essentially manipulate the microbiome in order to improve outcomes of patients with inflammatory bowel disease. And one of the best treatments we currently have for management of active Crohn's disease is exclusive enteral nutrition. I'm sure Sonia knows about that, and I'm sure some of you who are pediatric gastroenterologists know about that. But it's a very, very restrictive diet. So this is how much of this liquid-only diet children with active Crohn's disease have to consume to get better. Okay? So they have to consume approximately 120 liters over a period of 8 weeks to get better. So it's a very, very restrictive diet, but we know that it works very well. But one of the things we don't know about this diet is how it works. And in my team in Glasgow, we spend quite a lot of time and energy using next-generation sequencing, using metabolomic applications to try to understand how exclusive enteral nutrition changes your microbiome. And what we try to do, we try essentially to use this knowledge to develop new solid-based dietary therapies. These are some of the most important findings of this work. So we saw here very nicely that when these children get on exclusive enteral nutrition, their microbiome, which is already dysbiotic at baseline, which already has low diversity, gets even more suppressed. And as you can see, when these children are getting exclusive enteral nutrition, their microbiome community structure becomes already more dysbiotic, and guess what? These patients get better, their fecal calprotectin levels actually decrease. And this is how we used all this data in my group in Glasgow to develop novel dietary therapies which are based on solid food with the aim to manipulate the gut microbiome. So in other words, instead of giving children liquid-only diets, what we try to do here, we try to give them solid food-based diets which aim to induce the same effect on gut microbiome as EN. And these are some of the experiments we have carried out. But most important, we have shown clearly that if you use CD-treat, our new diet, you can improve disease symptoms, you can improve inflammatory markers, and so forth. And this is a very good example of translational research, and this is my last slide. So this is how we started in 2004 in Glasgow. These are the number of cans of exclusive enteral nutrition that children with active chronic disease have to consume to get better. This is how CD Treat looks like on the right-hand side, top corner. We try to intervene our patients by using what we call gold standard dietary methodology. We have a subcontracted catering company which prepares the meals. The meals are sent to the patient's doorstep. And believe it or not, this is how CD Treat looks when you ask one of the fine dining restaurants to prepare this for you. And with this slide, I would like to close my presentation, and I want to believe that in the future we'll see more of this application. We'll see more and more studies where we actually aim to change the microbiome in order to improve patients' outcome. Thank you very much for your attention.